Hello, my name is Dr. Roderick L. Roll, and today I will conclude the golden years. So as you see, we have been talking about the history of microbiology. We are currently on the golden years, and I broke this slideshow into two parts. This is the second part, which will address two more questions. So the first questions that we answered already was, is spontaneous generation of microorganisms possible? Then the next question was, what caused fermentation? Now we're going to look at the third question, what causes disease? Pasteur developed the germ theory of infectious disease in 1857. Since he knew that bacteria caused spoilage because of his previous experiments with the vintners, when he deciphered that yeast created alcohol and the bacteria created the acid or the vinegar, this helped him devise the germ theory. Pasteur later developed weakened strands for fowl cholera, anthrax, and rabies. These weakened strands would eventually be used as vaccines. So he named these weakened strains a vaccine. This led to the entire field of immunology. Immunology is a study of the immune system and how it controls and kills organisms that are foreign to the body. One of the first experiments we will address that looked at the question of what caused disease was the experiment where this scientist, Davaine, looked at an infection of cattle. He observed that in the blood of cattle, he found this organism. Subsequently, we know it's called Bacillus anthracis, but he found this rod-shaped organism that was in the blood of cattle. And he found that in 1850. In 1863, he inoculated a healthy sheep with those rod-shaped organisms that he isolated from the blood of the cattle. It subsequently caused the disease in sheep. He, however, did not win the credit for that experiment. Robert Koch did. So Robert Koch studied the causative agent of disease, and he received the credit for Davaine's work. What Koch ended up doing was discovering the etiology, what caused the original disease. So anthrax is primarily fatal to animals, but it can also cause ulcerations to the skin. This bacterium is called Bacillus anthracis, and this bacteria causes anthrax. So the etiology of anthrax is Bacillus anthracis. Koch discovered the disease in the patient, in the victim, and he discovered the disease in blood, sputum, or pus. He then took this organism and grew it on a potato slice. We currently now use stuff like gelatin or agar. So he isolated the organism from the blood by growing it on the potato slice. Then he infected another animal with the inoculum. That animal then developed the disease. When Koch performed those experiments, he had to create new techniques. Some of the techniques I will discuss now. So, laboratory microbiology advances were made by Robert Koch and others. Some are simple staining techniques. We're going to utilize a simple staining technique just to look at the shape of the bacteria. In this picture below, you can see the shape and the arrangement of shape just by performing simple staining techniques. So Koch and others discovered that. They also were the first to photograph or create a photomicrograph of bacteria. Number three, they also had the first photomicrograph of bacteria in a diseased tissue or in a diseased patient. Then four, techniques for estimating colony forming units per milliliter. And colony forming unit is the bacteria in a solution based on the number of colonies on a solid surface. We're going to estimate how many bacteria is going to be in an original solution. We're going to do a serial dilution in class, and then you're going to subculture some of the solution in petri dishes. Then you're going to count how many circles, which are the colonies, and you'll be able to estimate how many bacteria were in the original solution. So this was deciphered with Koch and others a long time ago. Another thing that these scientists found or discovered was they utilized steam to sterilize media. They also used petri dishes. They discovered aseptic techniques, and they saw that bacteria were distinct species. So Koch discovered what caused tuberculosis, and he won the Nobel Prize for it in 1905. The etiology of tuberculosis is Mycobacterium tuberculosis. So he disproved that TB was not inherited. Before 1905, people thought that you inherited this debilitating disease. How did he elucidate this infection and to know that it was caused by Mycobacterium tuberculosis? Koch created a series of steps to help him figure out what caused it. Those steps eventually were named the Koch postulate. So I'm going to now describe what the Koch postulate is. Step one, Mycobacterium tuberculosis is found in every patient with tuberculosis, but it is absent in healthy patients. So you take blood from both patients. It's not in the healthy patient. You don't see this bacteria, but you do see it in the diseased patient. Step two, Mycobacterium must be isolated and grown outside of the host. So you collect the blood, you collect the sputum, you collect the urine, and you subculture this organism on those potato slices or on agar. It has to be grown outside of the body. When Mycobacterium is introduced into a healthy patient, this healthy patient will succumb to the disease. They are susceptible. They will be infected by this disease, by this bacteria called Mycobacterium tuberculosis. That patient has to develop the same symptoms as the infected patients. Mycobacterium must then be re-isolated from the disease experimental host. So now the person that was healthy that you infected, you have to now isolate that same organism, the original organism, from them. That was the Koch postulate. This shows you how these organisms are named. We will discuss this extensively in class. We call it binomial nomenclature, naming organisms with two names. Right now I'm pointing at a very famous bacteria named Escherichia coli, and now you can see why it was named that way. It was Latinized after the original discoverer, and it has two names. The first name has to be capitalized, and the second name cannot be capitalized. And it has to be either underlined or written in italics. So now we will conclude with the final question. How can we prevent infection and disease? To address that, we're going to look at several experiments and several famous scientists. The first one is Dr. Semmelweis. Dr. Semmelweis was a medical professional, and he made his medical students wash their hands with chlorinated lime water. This is a disinfectant. And they had to wash their hands when they were leaving the autopsy room and going to the delivery room. Just by having his students wash their hands, he saw a reduction in women mortality during childbirth. So back in the day during this time, women were dying of a disease called puerperal fever. We now know that puerperal fever is caused by a Streptococcus infection. At that time, it was an 18.3% chance that a female would die in childbirth. Then, by washing their hands with chlorinated lime water, that mortality rate was reduced to 1.3%, meaning these women did not get the infection from giving birth. This was reduced by just washing hands. So this experiment was around the 1830s to 1840s. Unfortunately, Semmelweis died from a Streptococcus infection. Another scientist looked into how to prevent diseases also. It was believed that bacteria in wounds caused pus formation. So Mr. Lister thought it would be a good idea to spray the wound and the surgical incisions and the dressings with phenol. Phenol is a disinfectant. It is called carbolic acid. It has an intermediate to low killing capacity. And just by spraying a thin mist, he had a reduction of mortality by 2/3. And this occurred around 1870. In this picture, you see the guy to the far right. He is utilizing the spraying apparatus. So he is pumping this machine and it is spraying a thin mist where these guys are working. That reduced the mortality rate by 2/3. Another famous person was Florence Nightingale. She introduced cleanliness into nursing. Another person was Snow. Snow carefully documented an outbreak of cholera at a public water supply. This led to infection control in humans and the epidemiology to spread. So Snow was able to look at the spread of this disease that originated at this one watering hole. The disease, cholera, we eventually learned that it was caused by this bacteria called Vibrio cholerae. And Vibrio cholerae causes vomiting and rice water diarrhea. The etiology of cholera is that bacteria, and Robert Koch discovered it. He discovered the etiology of it. Edward Jenner also looked at how to prevent diseases. He observed that milkmaids rarely got smallpox because they were exposed to a mild form of it called cowpox. He subsequently inoculated a boy in 1796 with pus from a cowpox lesion. The boy developed an immunity to that strand and therefore did not get smallpox. This catapulted science into the field of immunology and the study of vaccinations. Edward Jenner knew of this practice from previously. This, in Western medicine, we say that vaccination started with him. But we now know that it originated in Asia and in Africa hundreds of years before. Finally, Ehrlich. Ehrlich was a scientist that envisioned discovering a magic bullet. Aniline dyes stained parts of the microorganism. So he envisioned if the stain was attaching to the bacteria, that somehow a drug could be anchored or linked to that stain, therefore targeting the microorganism and destroying the microorganism. So that was the magic bullet. So he subsequently searched hundreds of chemicals to try to find one that could destroy pathogens, but it was not toxic to the humans. After searching for those drugs, in 1908, Ehrlich discovered one. And he discovered this drug would target trypanosoma, which is the causative agent of African sleeping sickness, and Treponema pallidum, which causes syphilis. In this picture below, we are looking at trypanosomes. The squiggly worms are the trypanosomes, and the circles are blood cells, red blood cells.